Now we're going to look at chapter 13. We'll look first at invertebrates and later vertebrates. But before we can get that, let's just note that we're moving into kingdom Animalia. And we're going to be looking at representative animals and their characteristics for a few different phyla within kingdom Animalia, because there's over 30 phyla for this kingdom. Well, let's define first what is an animal. Well, animals have cells that are eukaryotic, they don't have cell walls. The organisms are multicellular and they're heterotrophic, meaning they eat other organisms in order to get their nutrients. We can also compare invertebrates to vertebrates. Invertebrates lack a backbone, and vertebrates possess a backbone. And kingdom Animalia is generally split into these two groups. About 95% of all animal species are invertebrates. However, you're probably thinking of animals that are vertebrates, like a giraffe, a rhino, a snake. Invertebrates actually make up the phyla of kingdom Animalia, or almost all of the phyla of kingdom Animalia, except for Chordata. So Chordata is the only phylum in kingdom Animalia that consists of those who are vertebrates. Invertebrates therefore vastly outnumber the vertebrates, and they live in most places, including aquatic and terrestrial or land habitats. If we look at phylum Chordata, that's going to include, again, the animals you're familiar with. Even though they're only about 5% of kingdom Animalia, they're things like fishes, amphibians, reptiles, birds, and mammals. And they're just the ones that we've come into contact with the most. We could also look at ways to classify animals that are not just vertebrate or invertebrate, like symmetry. There's bilateral, spherical, and radial symmetry. An organism with bilateral symmetry possesses a symmetry wherein if you cut it into two identical halves, you are able to do so via a longitudinal cut. Spherical symmetry is when an organism possesses a symmetry that creates two identical halves, but the cut can run through essentially the organism's center. And radial symmetry is when we see a cut create two identical halves by any longitudinal cut, not just multiple. But when we're looking at symmetry, we're looking at an organism and how it can be split into at least two identical halves. Depending on the type of symmetry, we might see that there are many other types of splits that can occur. So let's here look at bilateral versus radial symmetry. Bilateral symmetry is something that humans have, and when you create a longitudinal cut, you're going to have mirror images on the left and right. With radial symmetry, you can make a longitudinal cut anywhere along the central axis, and you'd be creating two mirror images. And then with spherical symmetry, not only can you do multiple longitudinal axi, you can do any in any direction. So it's a little bit more like more symmetrical radial symmetry. We can also compare anterior versus posterior, and anterior is towards the front, whereas posterior is towards the back. Now, if we're talking about something that walks on four legs, we tend to use words besides anterior and posterior to talk about towards the belly or towards the back, like dorsal and ventral, and then posterior would be towards the tail, or you might see caudal, whereas anterior might be cranial instead of towards the head. So it does depend on if the organism walks on two legs or four. But for most animals, we're going to be looking at they will have bilateral symmetry, and they'll have a region where their sense organs are, or a region that is towards the head. And when you picture yourself, this would be where your eyes, your mouth, a lot of your nerve cells are accumulating, and actually where your brain is located. And this concentration of sense organs and nerves in a particular location is called cephalization, and it makes distinctions possible between identifying ends of an organism. The anterior end always contains the head, the posterior end always contains the tail. Anterior also means kind of towards the front, and posterior means towards the back. And that again can change if we're looking at organisms other than humans. So here with this lizard, we are looking at anterior towards its head, posterior towards its tail. We could use something like cranial for anterior or caudal for posterior. And because this organism, the lizard, walks on all fours, we can also use things like ventral for belly and dorsal for back. Animals that have cephalization, they have an area that we would consider the head, tend to move forward using this anterior or head region. And that allows them to come into contact with parts of their environment and interact with them first using this cephalization or using their sense organs. Organisms with radial or spherical symmetry do not have this anterior posterior distinction, and we can't really see cephalization in those that have radial or spherical symmetry. It's only present in those with bilateral symmetry. A good organism to look at for cephalization is both humans and also cephalopods. Cephalopods have big regions we consider the head, where a lot of their neural network is located. And then humans have their sense organs at their head, including the brain, but also things like the eyes and the ears. We can also now look at some of the diversity of invertebrates. Let's look at some examples, like sponges from phylum Porifera. So phylum Porifera is characterized the most by sponges. They tend to live in marine environments, and there are some freshwater species, but most of them are going to live in seawater. Many are anchored and immobile, but not all. Some can move for a short period of their lifetime, but they all will initially and then eventually bind to a substrate. Substrate is just what they bind to. They do have two layers, an endo and epidermis, and the epidermis is the outer layer, and the endodermis is the inner layer of cells. And these are generated from the two germ layers. The endodermis in sponges is actually made up of collar cells, and these collar cells line the central cavity of the sponge. This central cavity is going to essentially act like both the stomach and also the excretion area. A jelly-like substance exists called mesenchyme or mesohyl between the epidermis and the endodermis, and it's not really cellular material, but it is necessary to the life of the sponge. We see this jelly-like substance be vital in the transport of nutrient material. A sponge is also supported by a network of spicules found mostly in the mesenchyme. They do provide a framework that supports the sponge. The spicules are made up of lime or calcium carbonate, sometimes silica, but it does depend on the species of the sponge. But these spicules are going to be made up of a tough protein called spongin, and they typically produce hard spicules. Spongin is a little bit softer than the harder calcium carbonate spicules, and the presence or absence of spicules and their composition also help biologists classify sponges. Sponge anatomy is viewable here in the image to the right. Notice the osculum at the top, it acts as an opening to the central cavity or spongocell. And we're going to see that sponges are filter feeders. They will take in food through pores on their sides called ostia, and the inside collar cells have cilia that beat around a central flagellum, along with the gel-like mesohyl and the flattened cells of the epidermis. We will see processes occur in the sponge that you might be more or less familiar with. Here are the spicules of the basket sponge with a hard calcium carbonate skeleton. Again, they're filter feeders. They must make their prey come to them, essentially. They are going to use their body and their many canals and cavities to intake water, and they'll push that water through their ostia and then up into their central cavity. The flagella of the collar cells also beat constantly, pulling water through the sponge as it passes through things like cyanobacteria, algae, organic debris. They will be extracted or captured and eaten by the sponge. They have specialized cells called amoebocytes that hang out in the mesohyl, and they use pseudopods to perform different functions in different animals. In the porifera or sponges, the amoebocytes travel freely in the gel-like layer between the endo and epidermis, and they're going to help extract food from what the sponge has processed. So again, filter feeders, they take in water through their ostia, and then they use their choanocytes and amoebocytes to process the material, but it will exit the osculum primarily. All right, next we can look at the amoebocytes in more detail. They not only help process food products, anything that is considered waste will be released from the amoebocytes. travel to the epidermis and then be released outside the organism. They can also function slightly in gas exchange, and they can communicate to the inner cell layers. So they're very useful. They can also help generate organization for the spicules that make up the bodies of some sponges. Once the food, however, has been extracted and digested, which is the primary function of the amoebocytes, water is expelled out the top or out the osculum, and then more is brought in. So sponges are very good filter feeders. Amoebocytes we can see here talking to this collar cell. They would take material that is absorbed into the mesohyl and help process it. Sponges also have different modes of reproduction. They can reproduce asexually by budding. They can also regenerate if some part of them is fragmented off. And they have periods of freezing temperatures wherein they produce a cluster of cells encased in a hard covering, usually spicule reinforced. And this gemmule is kind of like a cyst, and during periods of freezing temperatures, it is going to essentially hibernate. When the conditions are favorable again, the gemmule breaks open and a new sponge will grow from the encased cells. Here's what a gemmule may look like, and note the spicules protecting the inner membrane. This is asexual reproduction option in sponges. Essentially, it's protecting itself till conditions become favorable for it to persist. Sponges are also capable of sexual reproduction. Their sexual mode of reproduction occurs thanks to collar cells that can produce either eggs or sperm, and these gametes, when released externally, flow into the water, and this external fertilization can generate a zygote. Budding tends to be most common. Sexual reproduction can occur, but the dispersal or the creation of a gamete tends to be external, and then the free swimming larvae will find a substrate and develop into a new sponge. Now, you probably are most familiar with sponges that may be used for cleaning. Now, many of these sponges are synthetic, but there are some made up of these soft spicules made up of spongin that can be very absorbent and also useful in the home. Ancient Romans actually used sponges to pad the armor of soldiers. They've been used in painting, they've been used in surgical swabs, the fishing industry. So they're useful in many different ways besides household cleaning items. And that is it for our brief introduction into invertebrates. We'll move into part B next class.